Hello and welcome to another episode of Memento Now podcasts. In this episode, we'll be talking about and talking to uh, plant agriculture researcher, Dr. Alexandra Ficht. Would you care to introduce yourself?
Sure. Hi, Brandon. My name is Alexandra, and I'm currently a postdoctoral scientist. I'm working with Dr. Eric Lyons in the Department of Plant Agriculture. And prior to this, I did my PhD in the wheat breeding program at the University of Guelph. And so a lot of my research sort of dealt with the breeding program on a whole and how we can implement new breeding methods into our program.
Excellent, which brings us soundly into our questions. Would you like to give an overview of the goal of that paper during that PhD thesis?
Sure. So, as we all know, the advent of breeding is changing, and we now have access to more tools than we did in the past. So, when this project was given to me by Dr. Ali Nababi, his sort of goal behind it was to be able to merge our traditional breeding methods with newer breeding methods. And so, genomic selection uses both phenotypic data as well as genotypic data, and sort of merges those together to then provide new information about a breeding program, but bypass all of the field testing that we regularly do.
Oh, that's pretty impressive. So, what methods do you use to collect the genotype and phenotype data?
Sure. So, because with genomic selection, we're looking at updating our algorithms year by year. The reason we do that is because our breeding materials change year by year. So there's a couple of different methods. For our genotypic data, we actually went out and we took leaf tissue samples from the field, and then we did some DNA extraction, and we sent those out to the National Research Council out in Saskatchewan, and they actually did the genotyping for us. So what was unique about this project is that we used a newer genotyping method. It's called RAMP-seq, as opposed to the genotyping by sequencing or a 90K SNP chip that we've seen in the past. So for our genotypic data, we outsourced that. For the phenotypic
Can you go
data,
over the details of those three methods you laid out a little bit?
sure. So the difference is well, first of all, the reason we went with RAMP-seq is because I was using the entire breeding program. And so to genotype over 2,000 lines, it can get costly. So with RAMP-seq, what that method actually uses is our repeat sequences. The wheat genome is really large. And so we can actually utilize the repeat regions to then sequence lines. So it becomes a lot cheaper as opposed to genotyping by sequencing. Genotyping by sequencing, you get a lot more coverage, but with a lot more coverage comes higher costs, again, because our wheat genome is huge. It's hexaploid, so we have the A, B, and D genomes. And so it was just more of a cost thing than anything. Genotyping by sequencing is totally usable, and a lot of people do use it. And one of the advantages is that high coverage. But for RAMP-seq, again, just making use of the repeat regions was a lot more cost effective for us in this case.
So in this case, it's using specific regions in the genome that are common to all wheat varieties and looking at the variation between these repeats versus sequencing and genetically analyzing each individual plant all at once. Would that be correct?
Yeah, and across the entire genome. So the difference being that we would have a lot more SNPs on our genotyping by sequencing across the genome than we would for RAMP-seq. So RAMP-seq takes a reference genome, and within that reference genome, we have repeat regions that we would assume are present in all wheat varieties. And so we pick specific positions along the wheat genome and use those to then sequence, our 2,200 lines, as opposed to going across the entire genome and getting as much coverage and as much SNP information as possible.
Thank you for bearing with me on that not everybody knows about this sort of stuff, so I want to make this as available to as wide an audience as possible.
For sure.
Let's get back into the genotype and phenotyping methods. Sorry, I
Yeah.
interrupted a little bit.
No, that's okay. So that sort of covers the genotyping methods. Just a note, when we receive that genotyping data back, we do actually have to filter it for specific things like heterogeneity, for example. Another thing that's important is we had to filter out missing data. So with genomic selection, it's really important that we have really good genotyping data and really good phenotyping data. So in terms of phenotypic data collection, that was almost exclusively done in the field, except for some of our post-harvest traits. So some of the traits that I looked at were split up into two categories. We had agronomic traits, and then we had some disease traits. So for the agronomic traits, like plant height, that we just went out to the field to measure that. Yield, we take off the combine when it's harvested. We had some post-harvest traits like 1,000 kernel weight. So that's honestly just the weight of 1,000 kernels. We also had seed hardness. So there we have an SKCS machine, and that actually measures the hardness of the outer kernel. Those are important quality traits that people look for when they're developing varieties. And then for the disease traits, we looked at a wide range as well. Fusarium head blight. We did septoria, and we did stem rust, and leaf rust, I think. So all of those were actually visually scored in the field as well. And so just to note, some of the disadvantages with that is that we had to make sure that the same person was scoring each successive week across the years, because we didn't want any bias in our disease scoring. So often those traits are measured on a scale of 0 to 9, where 9 is highly susceptible, 0 is highly resistant. And again, the interesting thing with disease traits is that they vary year by year because they're very impacted by the environment.
Makes sense.
Yeah, so that was almost all of the phenotypic collection.
So you said you found a way to merge these two data sets together. So how did that happen?
Right. So in genomic selection, we sort of split our breeding lines into two groups. We have what we call a training population and a testing population. So what was unique about this project is because we were interested in the breeding program as a whole. So we made our training population up of our advanced breeding lines. So it may make sense if I kind of explain the flow of yield trials in our program before I jump into how we set up our training population.
It sounds perfect.
Yeah, so essentially we cross all of our lines indoors, and we carry those out to a specific homozygous percentage. And that way, when we go to grow our lines outside, we know that we're not going to get variation within a particular breeding line. So our first yield trial that we actually care about is called our preliminary yield trial. This is set up out at Aurora, and it's made up of anywhere from 500 to 1,000 breeding lines. And we grow those out, or we used to grow them out in single plots. So we basically gave each breeding line one chance to prove that we should advance it through the breeding program. From the preliminary yield trials, we then went into what we call our advanced yield trials. So we have two sets of advanced yield trials, or AYTs, AYT-A and AYT-B. And those are grown in multiple locations across 2 years. And so that gives us more of a chance to evaluate the lines in different environments across years. From our AYTs, we then go into our elite yield trials, or our EYT, and that's kind of the last chance for us to determine what we think may be a potential new winter wheat cultivar. So our EYTs from that point then go into some breeder trials if they kind of meet our quality trait expectations and the yield expectations, which are really important. And so for our training population, back to that, we took our elite and our advanced breeding lines and took our genotypic information and our phenotypic information for all of those lines, and we actually trained it on an algorithm. So essentially what that means is you take all of the possible information you have from a certain set of lines, and you allow this algorithm to sort of associate the genotypic information with the phenotypic information. So then when we apply the algorithm to the testing set, which is not included in our training population, we have a certain degree of relatedness between the two populations where we can, or the algorithm can deduce how they think that particular line will perform with respect to the trait that you're interested in. So hopefully that makes sense.
So using this algorithm, you can use the data obtained from the testing and the genotype data, and you can associate the genotype with the phenotype and predict how future versions of that same genotype will operate in new breeding lines developed from these varieties.
Yeah, exactly. That's pretty much the premise. And what's also important to note about the training population is that you want it to be representative of your entire breeding program, right? So you don't just want the best of the best. You also want some average performing lines and some poor performing lines so that when you test it on our preliminary yield trials, we can actually eliminate the crappy varieties without having to grow them in the field, right? And so that was the premise behind it, was cost effectiveness, time, like a time saving method to implement, because to grow 1,000 lines in a single trial at Alora is actually quite costly, and it takes a lot of time to phenotype, right? So if we could find a way to sort of bypass that phenotyping requirement, then that would be beneficial in terms of efficiency for the program.
Sounds pretty cool, actually.
Yeah.
So after you did all the testing, what conclusions did you draw from this paper?
Yeah, so there were quite a few. What we didn't know was how diverse our breeding program was. So the wheat program at Guelph is actually relatively young. It's actually less than 10 years old. So if you think about that in terms of diversity of plant material, the way that Ali actually started the program was to take material from different programs and from different companies and sort of use that as the basis for his program. So we ran into a few not issues, but hiccups, in that a lot of our training program or training population lines weren't super related to our testing population lines. So one of the conclusions we made is that perhaps genomic selection is better suited towards more established breeding programs. And that's just because you do need a degree of relatedness for the algorithm to do its job. Another conclusion we made is that instead of just using traditional methods or just using genomic selection, it's better to sort of merge both of those together, right? We don't want to get rid of our traditional breeding methods and just focus on algorithms, because we know that there are flaws with algorithms, right? Sometimes our degree of error is very high, and sometimes it may throw out breeding lines that would otherwise perform well. So it's still good to incorporate the phenotypic observations. Some other conclusions that we made was that the quality of your genomic selection output is dependent on how good your data collection is. So your genotyping method and your phenotypic data collection methods matter a lot. And so they need to be consistent across years and across environments. So to put it into perspective, we used 35 environments across Ontario, and sometimes it wasn't possible for someone to take the data in Woodsley, which is southwestern Ontario, and take the data in Ottawa at the same time because they were too far apart physically. So quality of data is really important. And I think those were kind of like the three big takeaways for that.
So would this data gathering be improved if they did in vitro selections like in vivo like greenhouse tests for disease resistance? Would that aid in making the data more consistent?
So there are pros and cons to doing greenhouse or growth room work as opposed to field work. Um, if we only because we can control everything, right, in a greenhouse or growth room setting to a degree, not 100%, but to a degree. What we found also, I didn't even mention this, is that we used G by E, so genotype by environment, as a covariate in our model. And so that was interesting in and of itself because across years, the environments change a lot. So um, we actually gathered really great field disease notes in 2019, I believe, because we had such high degrees of winter kill. So yes, it would simplify things, but I think that we're able to capture a lot more with respect to environmental impacts on diseases, for example, than we would in a greenhouse or growth room, if that makes
Yeah, it
sense.
does make sense. And from what I remember, I think that grow room testings have false negative, high level of false negatives.
Yeah, yep. And they kind of introduce their own disadvantages, right? Depending on if you're sharing the growth room with somebody else, things can be introduced. Sometimes the humidity and temperature have a huge effect on the plant growth and development. But it definitely has its place. But yeah, for genomic selection, having rigorous data, I would say. And you can kind of combine, for example, field phenotype data as with growth room or greenhouse phenotype data, and maybe it's even more robust.
Yeah. So another another thing was when it comes to the different weed selections, I actually did a bit of research into this, and surprisingly, there's a ever increasing level of genetic diversity within breeding programs coming out of Richtown. Would that be the same coming out of University of Guelph?
Yeah, for sure.
So I'm running out of questions actually.
That's
So
okay.
one more question. This is one I give to all my interviewees. If you could give any final messages to the audience, what would you give out?
Sure. I think, well, I had this experience when I was finishing up. Um, I think that the world of breeding is changing, and I think it's a really exciting time to be in breeding because we're kind of bridging the gap between, new knowledge and old knowledge. So I think that placing an emphasis on understanding both will definitely allow us to create more versatile or resilient varieties in the future. And I think that with variable climates, in the future, that it'll be really important that everybody that's involved kind of understands how the environment impacts what we grow and how we grow, but also how we can help producers. Because sometimes I find when I'm so focused on the academic research that we forget that we're doing this for a purpose, right? And the purpose is for producers and for society to benefit. So, yeah, that's kind of what I would say. Don't forget why we're doing it.
Excellent. You actually brought up a question. Do you mind if I ask it quickly?
Yeah, go ahead.
So when it comes to breeding more variable for more variable climates, is how would that be more of a quantitative trait thing where you breed for a large more adaptability to multiple climates, or would you breed more towards different different cultivars for different specific environments? Which way would be the best way to go for future breeders out there?
Yeah, that's a really great question. There's actually somebody working on that right now, doing her PhD. She's really awesome. Um, So the interesting thing with winter wheat is that we kind of have a unique climate across Ontario, right? If we were to compare that to Western Canada. And so by looking at some Western Canadian wheats versus our Ontario wheats, we've actually noticed a huge difference genetically between them, as well as the way they behave. But just to put it into perspective, if I was a breeder and I was breeding a winter wheat cultivar Canada wide, it would not fare as well as if I were to breed one for Southwestern Ontario, for example, versus Saskatchewan. The reason being, we have much different winter climates. So out west, it gets super cold. So the varieties that they develop for Western Canada have to be able to withstand a super low. They have to have freezing tolerance, right? Super low. In Ontario, we don't get as cold of temperatures in the winter, but we have other anomalies. For example, freeze-thaw cycles. So we've been seeing that this spring, actually, where we have a period of warmer weather, and then we had like a crazy snowstorm. And then we had another period of warmer weather and then another snowstorm. So the way in which we breed varieties for that environment is a lot different than for just an extremely cold environment. So we definitely need to be, it's not like we have to breed just for Elora, for example, but we can definitely focus on Ontario, for example, and then have the Western Canada focus on Western Canada.
Excellent. I think that about covers everything. Also, if you have any other graduate students or fellow graduate students that are interested in such a podcast, such as this, please let them know.
I will definitely do that.
And that was Dr. Fitch. Let's give her a round of applause.
Thank you, Brandon.
And I think that about covers it.
Thank you for sharing your time with me during this video. Please rate and share this video, and subscribe for more content. Thank you for watching.